Good morning. This is Dr. B. I hope you all are well. Starting Friday, tomorrow, we're going to begin remote instruction probably for the rest of the semester in all your classes. We're going to finish up Biology 102, uh, going through chapters 8 through 13. Chapter 8's topic is the role of mutation and genetic variation in organisms and the importance of mutation thus in the evolutionary process. So let's start out with definition of mutation. Some random change that occurs in the base sequence of DNA, something that changes a gene or an allele, alters the formation of a protein product. Some people consider mutations to be random. Some think that is not really appropriate. So, mutations are considered one of the prime sources of variation in populations of organisms. So, how do you think they occur? In some cases, they occur in interphase when DNA is replicating itself in the S phase of interphase, uh, making duplicate chromatids, if you will. Sometimes that mutation isn't repaired in the repair process after replication has finished. So the error gets transmitted on when the cell is reproducing. The error can occur during mitosis or meiosis. Perhaps a chromosomal mutation might occur when the chromosomes fail to separate properly in anaphase. So there's several different places within cell reproduction where a mutation can form. As I had begun this video, it is considered that genetic variation in populations is due to mutations, that mutations cause altered genes, and those altered genes or alleles form different protein products. And typically those mutations that are non-lethal are passed on. Lethal mutations can be passed on, and they do, but those that are non-lethal, that produce non-functional proteins, if you will, are often considered to be that source of genetic variation in populations. It is possible for a mutation to occur and result in no change in the protein product. It is possible for somatic cell mutations to be passed on in organisms that asexually reproduce. In organisms that reproduce sexually, it's primarily just the germ cells that pass on mutations to the next generation. I had told you that there are two basic types of mutations. One is called point, and the other is called chromosomal. The rest of this video will explain to you some of the different types of point mutations, and at the end give a few examples. Point mutations primarily occur when there are base substitutions, an insertion or a deletion of one to maybe two or three or four different bases within a gene. Typically, a mutation will have the most effect on a gene if it occurs in the first or second base of a codon. You will see in the genetic code that if a mutation occurs in the third base of a codon, those mutations are often called silent, and they do not have an effect on the eventual protein. We refer to this as the redundancy in the genetic code, that some amino acids are specified for by 2, 4, or even 6 different codons. So different types of point mutations. Some are called base pair substitutions. One base is mistakenly placed in during replication, and it's the wrong one. what might this have? What effect might this have on the eventual protein? One possibility is no effect. The same amino acid is coded for. Again, if the substitution is at the third codon of the at the third base of the codon, oftentimes there's no effect on the amino acid. That's called a silent mutation. So if the base pair substitution occurs in the first or second base of a codon, it can result in either a missense, the wrong amino acid being inserted, or a nonsense mutation where protein synthesis stops. A stop codon is specified for. It is possible for the reading frame of the gene to shift one way or the other, and that is called a frame shift mutation. These are often due to insertions or deletions. It is very possible for a frame shift mutation to result in a wrong amino acid being inserted. That's called a missense, or even translation or protein synthesis stopping, which is called nonsense. There are examples of each in the figure to the right of the text. So point mutations, just to go back, the substitution of one to several bases within a gene can form a different allele, some altered protein product, if you will. So examples of point mutations are insertions, deletions, substituting one base for another. It may result in a silent mutation where nothing changes. It may result in a missense mutation where the reading frame alters the amino acid sequence, or it may result in the protein process being stopped. Translation stops. That's called a nonsense mutation. So this particular slide talks about sickle cell anemia, where there is a point mutation where the amino acid glutamate is substituted with the amino acid valine in codon number 6. You notice the base adenine is substituted with the base uracil, and a different amino acid is inserted. That's a missense mutation, and this results in a different form of the protein hemoglobin, which results in some of the blood cells being deformed, and their capacity to carry oxygen is reduced. So we refer to this pleiotropic condition as sickle cell anemia because it has a multitude of effects on the human body. A second example of a point mutation is cystic fibrosis. It is the result of a deletion of a codon that has an effect on a transmembrane regulator protein that's involved in the functioning of cilia. So cilia are important in the removal of mucus from the bronchial tubes, and without proper functioning of the cilia, thick deposits of mucus can develop. And let's see about this on the next slide. So, as you know, the first picture, a healthy lung cell has this membrane CFTR protein that allows chloride ions to pass freely, and this results in normal functioning of the cilia. Cilia beat back and forth. Think of them as almost like your eyelashes. Without this proper functioning protein, and you see there are two possibilities. Number one, the protein is formed, but it can't open and close to allow the chloride ions to be released from the lung cell. So the cilia do not function properly and thick mucus develops. The second mutation actually results in no protein forming at all. So there's no way for the chloride ions to exit the lung cell, and this mucus deposit occurs as well. So those are two different mutations that can occur in a gene with this transmembrane protein. And in both cases, the symptoms of cystic fibrosis occur. There's one more example that I wanted to show you of a point mutation, and this is called Tay-Sachs disease. It's due to an altered protein called hexosaminidase. Without this properly functioning enzyme, nerve cells will begin to degrade, and oftentimes a child will die before they reach the age of 5 due to the degradation of their central nervous system. So this particular point mutation is due to an insertion of 4 base pairs, which causes a very extensive missense mutation. So I've given you 3 examples of point mutations: sickle cell anemia, cystic fibrosis, and Tay-Sachs. So this is kind of the end of this first video. In the second video, we'll talk about chromosomal mutations and give some examples of them. Thank you. God bless. Have a great day.